experimental design by Arjun Rambia. So the question we have is, how can the scientists deduce which of the 5 randomly selected organisms has MELAS syndrome, which is caused by a dysfunction in the mitochondria? So some background, the mitochondria converts glucose into chemical energy called ATP that is needed by the cell. People with MELAS syndrome, which is also known as mitochondrial encephalomyopathy, lactic acidosis, and stroke-like episodes syndrome, have a high buildup of lactic acid in their bodies and, as a result, have increased acidity in their blood. And MELAS syndrome is caused by a mutation in the mitochondrial DNA, which is also known as mtDNA. So our hypothesis is because people with MELAS syndrome have increased acidity in their blood, if the scientists gather each of the 5 patients' blood samples and then test the blood pH, the person with the lowest pH, which will also be the most acidic, will have MELAS syndrome. So the materials needed, we need blood samples from each of the 5 subjects, and then we needed a tourniquet, a blood drawing needles, and blood collection tube to get the blood, and then a glass electrode to measure the pH of the blood. So our independent variable is the blood sample from each of the subjects. The dependent variable is the blood pH level. Control variable would be a blood sample with the normal blood pH level, which is about 7.36. And our constants are room temperature, the glass electrode we use, and the method in which the blood sample is taken. So first, we need to ensure that each of the patients do not eat or drink anything for 8 hours prior to the blood test, so the results aren't skewed. And then we need to gather all necessary materials, and then draw a sample of blood from the organism/patient using the tourniquet, blood drawing needle, and the blood collection tube. And then we can use the glass electrode to measure the pH of the blood sample we just took, record the data, and then repeat steps 2 through 5 for the next 4 organisms. So after we gather all of the blood samples and measure the pH levels of the blood samples using the glass electrode, we can compare them to each other and the control. Again, the control would be the normal blood sample that has a pH of about 7.36. So anything lower than that would be considered acidic for blood. And the organism who has the blood sample with the lowest pH, which again is the most acidic, will have MELAS syndrome.